Hey, welcome back. This is Mr. P. We are going to further our discussion of evolution now by specifically talking about how evolution of a species impacts the population as a whole. And if you remember from last lecture, we talked specifically about how individuals can't evolve, but populations can. In my lifetime, I will not change as a species. I can adapt to my environment. I can change environments and become better adapted or better suited, more well equipped for my particular environment, but I'm not going to change as an entire species. I was born a human, I will die a human, and all other organisms are the same way. But populations as a whole can change over time, and that's really the premise of what evolution is. And so some background on exactly what evolution is. We're talking about DNA at a basic level. DNA is the blueprint that makes up life, and DNA is universal in nature. Okay, we talked in chapter 12 at length about how the universal code, that is alternating A, T, C's, and G's, and how those come together to form a double helix that basically gives the instructions in our genes on how to live and how to produce an organism. There are pieces of evidence or pieces of work that have happened since Darwin's time, and Gregor Mendel, who was instrumental in the discovery of mathematical probabilities related to genetics and the potential outcomes of a genetic cross. His work and Darwin's work connected in the 1930s. And because Mendel, who was instrumental in talking about genes and DNA and predicting probabilities, that was really good. That was a really good piece of evidence that connected Darwin's common ancestral theory together. Okay, organisms have heritable DNA. They have heritable genes. Those genes will interact as an organism's population goes on into history or into the future, and depending on how organisms pass on those traits will dictate where the population goes in the future. Okay, changes in genes produce heritable variation. It's this heritable variation, often through mutations, that results in new characteristics. Those new characteristics will either be selected for or selected against, and that's when we get into this whole idea of natural selection. Okay. Natural selection is environmental pressures that are placed on traits that are either are positive or negative. And if those traits are positive, then those organisms are going to be more fit in their environment, and they're going to pass on those organisms or those traits to future generations more readily because they're going to reproduce more. Those organisms that have unfavorable characteristics are going to be least fit or less fit in their environment, and they're not going to reproduce as much, leading to a decrease in that particular population, okay, or a decrease in that gene or trait in that population over time. Evolution works through variation. You need to have variation in order for evolution to work. If you didn't have variation, there would be nothing to select for or against, and there would be no driving force for this idea of evolution. Watson and Crick were the two guys that looked at Rosalind Franklin's X-ray crystallography images and were instrumental in developing the first three-dimensional model of DNA. It was their model and their demonstrations that kind of connected this molecular DNA structure to the variation, and then ultimately to Darwin's work. So how common is genetic variation? Remember when we talked about genetics, when we talked about heterozygosity and homozygosity? Heterozygosity, remember, is the condition of having two similar, or two rather different alleles, one big and one little, like big A, little a. And homozygosity is when you have two of the same alleles, like little a, or big A, big A. So heterozygosity is different for all organisms, but we are mammals. And when you look at mammals, fish, and reptiles, only about 4 to 8% of their genes are actually heterozygous, meaning that the majority of us, in fact 92 to 96% of the mammalian genotype is homozygosity, okay, or homozygous. There is much more heterozygosity in insects, which is why there's so much more diversity in the insectual or insect populations. Okay, 15% of their genes are heterozygous as compared to only 4 to 8% of mammalian genes. How does this variation lead to this idea of gene pools? Now, if you remember from your vocab, gene pool is basically how many genes there are in a particular population. Okay? Gene pools are all the genes or alleles present in a population. Okay? And there's a typo. There should be present in the population. Depending on how many genes you have in the population and how many different alleles you have in the population will dictate how much variation you have in the population. The more diverse the gene pool, the more variation you'll experience or observe in that particular population. Frequency is the number of times that an allele occurs in a gene pool. Okay, so how often is this particular gene present in this particular gene pool? That is frequency. Okay. Evolution is a relative change in the allele frequency in a population over time. This is basically the definition of evolution. When you talk about the frequency of an allele in a population over long periods of time, for the most part, is evolution. Okay, so evolution is usually seen as this really scary kind of abstract concept, but really at the basic level, if you change the frequency of a particular gene in a particular environment or a particular population over time, it is a change in that population over time, and so evolution has happened. Sources of genetic variation include mutation most of the time and gene shuffling some of the time. Okay, mutation. How do most genetic variations happen? A lot of times mutation is to blame. Okay, mutations can cause favorable characteristics or favorable changes. Sometimes they'll cause unfavorable characteristics or changes. Gene shuffling would be the sexual reproduction part of genetic variation. Okay, when a male and a female have a baby, they are essentially shuffling the gene pool, so to speak, because they're passing their genes on, or at least a certain portion of their genes on. So how does mutation affect variation? Remember, mutation by definition is any change in the genetic sequence of DNA, mistakes during replication. Okay, we've talked at length about DNA replication. Remember, it's a semi-conservative process, meaning half of the old DNA serves as the template strand to produce a new complementary strand. But there are mistakes that can happen, okay? Just like any typo can happen. We do have enzymes that act as spell checkers, and for the most part they do a pretty good job of checking for errors and fixing those errors, but they're not always 100% efficient, and sometimes mistakes happen. Any mistakes during the replication portion obviously are called mutations. Now those mutations can be, like I said before, beneficial, or they can harm the organism, or they can be neutral. Okay? A neutral mutation is a mutation that happens but does not affect the fitness of the species at all, because it was probably a mutation that happened in the intron portion of the DNA, not necessarily the exon. Remember, the exons are the portions of DNA that will actually become expressed as proteins. Some of the mutagens, or things that cause mutations to happen, can be things like chemicals, radiation, etc. Okay, some mutations are silent. Some mutations affect fitness. Like I said, they can be positive or negative. Silent mutations would be things like neutral mutations. Okay, if you change a UGG to a UGA, so to speak, chances are it's going to not change the amino acid, and so it won't change the protein, and so it won't actually show up in the organism's genotype, and then it won't show up in the organism's phenotype. But yeah, mutations. So how does gene shuffling impact variation? So gene shuffling most of the time occurs during gamete production. Now, why do we produce gametes? Remember, gametes are sex cells, and so sperm cells and egg cells are the gametes. Those gametes have to come together during copulation, or during sexual reproduction, and those gametes were first produced by a process learned 2 chapters ago called independent assortment. Remember, independent assortment is how chromosomes line up randomly and then are distributed randomly. Crossing over can happen. Crossing over, remember, is when 2 homologous chromosomes come together during prophase 1 of meiosis and can exchange portions of their arms and exchange genes that way. Okay, they can, just in this crossing over, produce drastic phenotype differences, and that's one way to shuffle the gene pool. Also, sexual reproduction, like I talked about before. Sexual reproduction, remember, is recombining different individuals' gametes. Okay, all people are different because all people were born to different people. Even the same people having multiple offspring are going to have multiple different versions of their genes, just based on the way the chromosomes line up and separate, and also random crossover events. Okay, all of these things help to shuffle the genes. Why do we want to shuffle the genes? Because we want to increase variation. Why do we want to increase variation? Because variation is good to keep a population competitive and moving forward. We want variation so that we have a greater chance of randomly acquiring favorable characteristics. If we randomly acquire favorable characteristics, then we're going to weed out the unfavorable characteristics, and we're going to let the strongest characteristics rise to the top. Okay, we call that survival of the fittest. An analogy I like to use when we talk about gene shuffling is the deck of cards is always kind of the same deck. Okay, we always have the same 52 cards. Sexual reproduction is shuffling the cards. Okay, we can shuffle the cards all the time. That is sexual reproduction. We are taking two unique individuals, we're shuffling the deck, and we're pulling a hand out of that. Different hands can be achieved by pulling 4 cards from that deck, but the allele frequency did not change. Okay, the allele frequency is the number of kings in that deck, or the number of 2s in the deck, or the number of 10s in the deck. The allele frequency didn't change, but pulling out the 4-card hand can change due to shuffling. Okay, the more we shuffle it, the greater the chances we have of pulling out a 4-card hand that differs from the previous 4-card hand. Okay, if you never shuffled your cards, you're probably going to pull out the same 4 cards, put them back on top, pull out the same 4 cards, put them back on top. Okay, we want to shuffle the deck. Now we're getting into this idea of single gene versus polygenic traits. Now I know when we did genetics, we talked about polygenic traits. Remember, skin color is an example of a polygenic trait. Hair color, human height, human weights, eye color, all of these things are examples of polygenic traits. We talked about how many different phenotypes there are as a result of polygenic traits. There are a lot of human heights. We don't have a tall version and a short version like pea plants do. Single genes are things like when we talked about pea plants, and we did just basic monohybrid, dihybrid cross stuff. Pea plant height is a single gene trait. You either have a tall plant or a short plant. In humans, the widow's peak is a single gene trait. Okay, widow's peak is whether or not your hairline in the front kind of forms a V. That's controlled by a single gene. You either have it or you don't. Polygenic traits, on the other hand, the more dominant traits you have, the more of one color your eye might be. So the more dominant of the eye color traits, the more brown your eyes, and the more recessive, the more blue your eyes are. Okay, so if we look at single gene traits, like I said, widow's peak or no widow's peak, that is a single gene trait, only 2 distinct phenotypes with 1 gene traits. Okay, it's usually you either have it or you don't. Allele frequencies may not match Mendelian ratios. Okay, allele for widow's peak is the dominant allele, however, doesn't have to be the observed the most often, because there's no advantage when we talk about survival of the fittest for those individuals that have widow's peak and not have widow's peak. Okay, when we talk about Mendelian ratios, remember that is more of a best case scenario kind of theoretical value. We don't necessarily always follow those ratios in real life, but when we look at a single gene trait, if one of the other, okay, for instance, if widow's peak allowed us to survive, you would expect, or allowed us to survive better, you would expect to see a higher percentage of the population with widow's peak. If no widow's peak allowed us to survive better, you would expect to see a much higher. Now I know there is a higher percentage of the individuals without it. That's probably just due to other factors. Okay. If we look at polygenic traits, you will see more of a bell curve distribution. Okay, traits controlled by 2 or more genes. because you are controlled by 2 or more genes, you will see a distribution of phenotypes. There are multiple phenotypes. Now, human heights are a phenotype, okay? Your phenotype at 70 inches as a man, you would be the average phenotype. Now, you could be 65-inch man, you could be a 60-inch man, you could be a 55-inch man, you could be an 85-inch man. You have one specific height, so you have a phenotype, but there is a distribution of heights in a population. And so there are individuals that are 65 inches, and there are individuals that are 80 inches. The majority of the human heights in men congregate right here around 70 inches. Okay? Same thing for women. Now there's a higher peak because there's more women in this average than there are men at the average. There is a greater spread out, or a greater range of heights in men than there are women. Okay, now we talk about natural selection. Natural selection does not act directly on a gene. Evolution acts on the population but affects the individual. Remember, evolution impacts population because we are impacting the frequency of genes in a particular population over time. Evolution does not affect the individual, or I should say doesn't act on the individual but can affect the individual. Okay, the gene does not survive or reproduce, the organism does. Individuals with adaptation must reproduce to pass the gene down. Those that reproduce the most will pass that gene down more often, leading to a population shift. The gene is changed, but the individual passes the gene to the next generation. Populations evolve, individuals do not. Okay, I said that's going to be one of the most important keys to this chapter. Populations evolve, not individuals. So, natural selection on a single gene trait. Natural selection on single gene traits lead to changes in allele frequency over time. And if you change the allele frequency in a population over time, you have evolved that population. Changes in allele frequency over time occurs because there is something about that change that enables an organism to survive and pass its gene down to the next generation. Again, we talk about organisms that are the most fit are going to be the most successful at passing their genes down. And if they are the most successful at passing their genes down and they reproduce the most, they're going to change the population to be more like them just based on the number of offspring in the population that resembles them. Okay? If you look at this, lizard color, we've got 10% of the population, initial population, notice initial population, 10% are red, 10% are black, 80% are brown. After 10 generations, we go back to this individual ecosystem or individual location, environment, and we make other observations. 80% are still brown, so the brown population is holding steady. Now, the black lizard population has doubled from 10% to 20%. The red lizard population has decreased to 0%. So red lizard extinct. No more red lizard in that particular area. After 20 generations, the brown lizard population is slowly decreasing, and after 30 generations, the brown lizard population is even decreasing again. Okay, what does that tell you about our environment? The most fit organisms are black. How do you know? Because the black population is increasing over time. In fact, it basically doubles every 10 generations. We go from 10 to 20, 20, almost to 40, 40 or 30 to 60. Okay, we double about every 10 generations. Here we're going down. So which color or which population is the most fit? Black lizards would be the most fit. What does it tell you about the environment? You probably have black soil or dark black colored rocks. How does natural selection affect polygenic traits? Okay, the effects of natural selection is more complex due to the range of phenotypes capable of being expressed. Remember, in polygenic traits we have a range of phenotypes like human height or human eye color. And so fitness close to one another on the curve may not vary greatly, however the ends of the distribution may be affected more. Okay, on the distribution curve I showed you before, there was a big range or a big difference between those organisms that are, say, 50 inches and those organisms that are, say, 90 inches. Okay, if you were able to survive better at 50 inches, then you would expect the curve to kind of shift down towards the 50-inch range because the 50-inch people would be making or having more babies and leading the population to change towards them. Okay, natural selection can affect the distribution of phenotypes in 3 ways. This is really important. Those 3 are vocab words: directional selection, stabilizing selection, and disruptive selection. Okay, and now I want to talk about how each of those look and what has to happen in order for those 3 selection types to happen. Okay, the first one, directional selection. If this is our original population and we're looking at rock pocket mice, like the video we watched, okay, rock pocket mice, we have mice that vary from very light colored fur to very dark colored fur. Notice that there are not very many dark color, okay, a very low population of dark color and a very low population of light color. What would this tell you about the original habitat? Probably the habitat is kind of medium brown, because the medium brown fur has been or has become the most successful. Okay, it reproduced the most, it lived the best, obviously leading the population to average out at about the medium brown. Although, if a change in the population, okay, or let's just say a change in the environment, and let's say the rocks got more black or lava flow came through just like in the video we saw, and all of a sudden now the darker fur is starting to reproduce most, okay, or starting to reproduce more, and the light colored fur reproduces less, then the original population curve shifts to the right, because now our average is this color and not this color. Okay, that is a directional selection. We are shifting the curve one direction. Now we can either shift it to the right or we can shift it to the left, but either way you are shifting the distribution curve one way. If we look at stabilizing selection is exactly what it sounds like. We're stabilizing the population. If our original population runs from very light to very dark, with our medium brown mouse being the average, if our population doesn't actually change, or our environment doesn't actually change, but there's a greater environmental pressure placed on the dark and light, then those 2 populations are going to die or decrease, leading to a pinching in and a greater population around the average. So our average doesn't change. We still have the exact same average, but we increase the population of our average and we decrease the populations of the 2 extremes. So the 2 extreme populations are selected against, the average is selected for. That is stabilizing selection. Our curve has stabilized at the average. And then we have disruptive selection. Disruptive selection is the selection where the 2 extremes are now favored, okay, but our average now is favored against or selected against. So our medium brown mouse is no longer the most fit, okay, it used to be, but let's assume now that we have an environment that once was kind of light sandy brown sand, and all of a sudden white and black snow and rock, or light colored rock and dark colored rock, come in and cover the sand. Now all of a sudden the white colored mice can hide on the white background and the black colored mice can hide on the black background, but the brown colored mouse can't hide on any of the backgrounds. So all of a sudden now the predators are feeding on the once highly populated brown mouse, and now the 2 that were not selected for are selected for, and so we now have kind of 2 peaks on our curve. Okay, this is what leads to speciation. If this drops to basically 0 and these 2 mice populations no longer breed with each other or no longer recognize each other as the same species, then speciation can happen and we could end up with 2 distinctly different species. This just shows both. Okay, we kind of summarize. Again, we have an original population. If we slide our original population one way, that is directional selection. If we stabilize by pinching in the 2 extremes, it's called stabilizing selection. Again, we still have the same average. Here our average shifts to the right or the left. And if our average decreases but our two extremes increase, then that is disruptive selection. That leads to speciation. If we talk real briefly about this idea of genetic equilibrium and a Hardy-Weinberg principle, which is not something that you're going to really need to calculate or understand for the test, but it's used to kind of look at or calculate the allele frequencies within a population by looking at constants and specific factors that causes those frequencies to change. Genetic equilibrium is one of the factors that plays into this idea of Hardy-Weinberg, and conditions that are required to maintain equilibrium from generation to generation have to be factored in as well. One thing that Hardy-Weinberg principle is vital about, or important, one of the important components to think about, is this idea of random mating. Now, random mating does not happen in most populations because it is nearly impossible. Populations don't mate randomly. There are always things or characteristics that mates are selected for, okay, or some criteria. If you think about in white-tailed deer populations, those white-tailed deer that have the biggest antlers and are obviously able to fight off other males win breeding rights. Same thing with a lot of other species. There are a lot of male species or male organisms of species that are going to fight for mating rights in a particular environment. Okay, and so that would not be random mating. Random mating is literally you kind of just put all organisms into a hat, pull out names, and say go, or so to speak. Okay, non-random mating means some traits are under the some strong selection pressure, but not others. Genetic drift is an important component of evolution. Okay, so another form of random change in allele frequency that occurs in small populations, not due to natural selection. Remember, natural selection is when nature selects for particular traits. Individuals carry a particular allele in small populations that may reproduce at a higher rate than the rest of the population. Over time, a series of chance happenings could cause the gene to occur at a higher rate than normal. This happens when a small group of individuals colonize a new habitat. They have a different allele frequency than their original population. That is called founder effect. So basically what is happening is if we have a large population of humans in North America and one very small selective group of individuals that all have very similar traits, if they're picked up and moved to Antarctica, for example, and they start a new kind of colony at that new habitat, that small group of individuals is not going to have the same kind of allele frequencies that the big population of the world or the big population of the United States or even North America would have. Okay, so small populations, small niche groups of populations can have and often do have different allele frequencies than their original population. Okay, and so that's called founder effect. Evolution versus genetic equilibrium. One thing that is vital is that populations must be large. Genetic drift has less effect if populations are large. If populations are large, they can withstand immigration into and immigration out of because they have a large sample size. You're not going to change the population allele frequencies very much by moving certain individuals in and out. If you have a small population, then it's more detrimental to the population to move individuals in and out. Okay, so evolution, population must be large, no movement into or out of the population. Immigration or immigration. Immigration is individuals exiting a particular environment. Immigration is organisms moving into a population. Okay. Populations' gene pools must be kept separate from another population gene pool. No mutations can happen. No natural selection can happen. No phenotype should have a selective advantage over another. Now we know this is kind of best case scenario. It never happens like this because not all genotypes must or are going to be shown in equal proportions. This is kind of a theoretical best case scenario type scenario. Doesn't always work like that. Now, if we talk about isolating mechanisms, 3 different isolating mechanisms: behavioral, geographic, temporal. Temporal isolation, geographic isolation, and behavioral isolation are 3 ways that cause species to isolate themselves in particular environments, and we'll talk about each of those. So behavioral isolation. Behavioral isolation is when 2 populations are capable of interbreeding but have differences in courtship rituals or birdsong or other reproductive strategies that involve behavior. So if it is a behavior that keeps 2 potentially mateable species separate, it is a behavioral isolation idea. Okay, example: Eastern and Western meadowlark. They don't respond to each other's songs. They do not interbreed, even though they are capable of interbreeding or breeding with each other. They could reproduce together, but they choose not to because they have different songs, different courtship rituals. That is a behavior that keeps them separate. If it is a behavior that keeps them separate, that is behavioral isolation. If it is a geographical barrier or a geographical item that keeps 2 species separate, then it's geographic isolation. So 2 populations are separated by a geographical barrier, such as a river, mountain, or ocean, bodies of water, then it's geographical in nature. Now, a river might be a geographical barrier to 2 species of field mice, but they wouldn't, or that river wouldn't be a geographical barrier when you talk about birds, because birds can just fly over the river. A mountain range is a pretty good geographic isolator when you talk about birds, as well as other things. Okay, very few organisms can cross a mountain range to go and interbreed with the population on the other side. So, an example of geographic isolation includes 10,000 years ago, the Colorado River separated the species into 2 separate populations, and we're talking about these 2 particular squirrels. These are different squirrel species. They are living on opposite sides of the Grand Canyon. You do know that the Grand Canyon was formed by the Colorado River, and because the Colorado River started cutting through their habitat, which was once the same species habitat, it kept them apart, and they no longer interbreed with each other because they can't physically come back in contact with each other. Okay? Geographic barriers do not guarantee the formation of new species because things can happen. Okay, a river can run dry and organisms can cross at that point. Lakes can flood, and so organisms that are in a body of water can then go to another body of water and interbreed with fish of other species, if you're talking about fish populations. Okay. This is talking about the range of the squirrel. Everything in pink is the range of the Albert squirrel. In purple is everything the Kaibab squirrel. Notice the Colorado River that runs right through this range cut the range of this squirrel in half, and I know there's a lot more of it here than there is here, but this is the only place you find that particular species of squirrel, very closely related to this species, only geographically isolated. They cannot cross that river or the Grand Canyon at this point. I think that's where we're going to leave it. That is the end of our evolution discussion. Make sure you bring your questions and notes to class. See ya.